Okay, good afternoon, everyone. Just before we get started, I'd like to do a declaration of interest. None of the speakers in this session have financial disclosure or conflicts of interest arising from these presentations, and the British Society of Nanomedicine has received an honorarium from Viv4 Pharma for the delivery of these presentations. So today's session is a series of 3 talks. We're going to be talking about from cancer to COVID, 25 years of nanomedicines. We are all trustees of a charity called the British Society for Nanomedicine, which I'll give you a bit more information about shortly. So I'm starting the talks today. I'm talking about the history of nanomedicine. I'm Tom McDonald. I'm a senior lecturer in nanomedicine from the Department of Chemistry at the University of Liverpool, and I'm also co-chair of the British Society for Nanomedicine. Just before we get started on my talk, I wanted to just give you a bit more information about the British Society for Nanomedicine. The Society was created just under 10 years ago to create a focal point for researchers in the UK across academia, industry, government, and also within regulatory authorities. And we also seek to engage with the public in terms of new scientific and commercial developments in the field of nanomedicine. Specifically, we have 4 other aims. One is to educate the public, also to provide a forum for the development of future researchers, to represent UK researchers outside the UK, and also try and connect in with other European and global societies. So hopefully these talks will give you a greater interest in nanomedicine, and I'd suggest if you do, you go to our website. The link's just on the bottom right of this slide. That has a lot more information and will help keep you updated. I'd also encourage you to join the society. It's completely free and allows you to keep updated with any activities in the space of nanomedicine. So my talk, I'm going to give a bit of background about nanomedicines. I'm going to talk about the history of nanomedicines, and then I'm going to look at 2 specific early examples of modern nanomedicines. So before we do that, I think it's very important to obviously have a clear definition. So there are actually a range of definitions used for what is a nanomedicine. The one I personally prefer is this one given here. So it's nanoparticles for medical applications with sizes between 1 to 1,000 nanometers. So that's obviously a huge range of sizes within that, from 1 nanometer up to 1,000. So just to give you a better feel for that, there's this picture shown on the slide. So in the middle, you can see a little ring that is 1 micron. And you can see that that 1 micron or 1,000 nanometers is smaller than things like bacteria, chromosomes. It's just slightly larger than the wavelength of light. And some virus particles are just below this size. So at the top end of the size range, we're looking at very complex structures. When we go down to the bottom end of the scale, we're approaching the sizes of many molecules. So you can see the circle in the middle for 1 nanometer. Glucose molecule is just smaller than 1 nanometer. Phospholipids, the lipids that make up our cell membranes, are larger. And then also some other molecules or structures can be larger than this. So this scale is very broad, and it goes from essentially macromolecules all the way up to complex multi-component structures. So why is this an important size range? Well, if we think about it in a rather simplistic way, it's large compared to most molecules. So most nanomedicines are tens or hundreds of nanometers in diameter. So these are much smaller than the molecules that are typically used in traditional therapeutics. So you can't really see it, but there's a cartoon of the phospholipid bilayer, and above that there's a molecule which is sucrose. And you can see that a 20 nanometer particle is huge compared to the biological structure of the membrane and also the molecules. So its behavior is going to be very different from traditional small molecules. The flip side of that is that it's actually nanoparticles or nanomedicines are small compared to biological structures. So the big red thing on the right is a red blood cell. And then we can see on the left of that blood cell in the box is some schematic representations of nanoparticles of different sizes. So there we've got nanoparticles of 200 nanometers, and then within the smaller box we can see even smaller nanometers that might be, say, 20 nanometers. So it's a very so these are much smaller than some of the structures that are present in our bodies, like cells, and so this triggers very different behavior. So what do these nanoscale properties offer in terms of drug delivery opportunities? So there's two, and I'm going to quickly give a bit of an overview. So one is about adjusting the biodistribution. It's about having the ability to potentially target a disease. So as we can see in the cartoon, we've got a localized disease shown in red, might schematically be represented. It might represent cancer, for example, a tumor. And systemic dosing is obviously going to treat the whole body and going to lead to additional side effects in off-target sites. However, if that can be targeted to localize into a site, we can improve the patient outcomes. So this is typically used for an IV dosing approach. For another opportunity that's achieved through this nanoscale properties is the ability to improve the pharmacokinetics. So this might be evening out patient variability or improving the oral bioavailability of a drug that's being dosed that might have low aqueous solubility. I'm not really going to go into huge detail on this, but another opportunity is to potentially open up different routes of administration. So you might have an oral formulation that you could make long acting, or you could formulate something that so it's got improved topical penetration, or also change the ability for something that's inhaled, for example. I'm going to give two examples, one that fits within the oral dosing approach and one that's intravenous at the end of my presentation. So let's have a look a little bit more about why these nanomaterials can offer benefits. So in terms of targeting, in terms of changing the biodistribution, the ability to encapsulate the drug's key. So this picture shows quite a nice representation of a nanoparticle. Inside we've got some the therapeutic, and on the outside in this case there's some targeting ligands. So the drug's contained. That ability to contain it allows drugs that are very toxic, like cancer drugs, to be contained until potentially they reach the tumor site. Whereas conversely, drugs that are very sensitive can be contained and protected until they get to the target site so they're not degraded. So in both ways, you're improving the efficacy of the treatment, changing the biodistribution. Another key one is altering the biodistribution. I'm not going to have detail to go into this, but the size can lead to some accumulation, either passively based on filtering mechanisms, or the surface binding can also improve targeting and biodistribution. So here's a schematic from a paper that shows in red free drug and in green the nanoformulation. And you can see that in the case of the nanoformulation, the clearance rate in blood is decreased, so the graph on the left. And if we look at the graph on the right, the accumulation in the tumor is enhanced. Some of the off-target effects, so in the kidney, heart, and brain, are also reduced for the nanoformulation. The other way, so when we're talking about PK, is a bit more simplistic, but equally effective, is simply about increasing surface area. As shown in the picture on the top, if you have a 1 centimeter cube particle, it has a surface area of 6 centimeters. If you break that down into 1 millimeter cubes, its surface area will be 60 centimeters squared. And if you break it down into 1 nanometer cubes, its surface area will be 60 million centimeters cubed. So by breaking things down into smaller objects, we massively increase their surface area. What does this do? Well, increasing surface area increases rate of dissolution. So we know this practically in day-to-day life. If you want to put not tea in your drink, if you want to put sugar in your drink, you put a sugar cube in, it's going to take longer to dissolve than granulated sugar, or if you're so inclined, if you want to get other icing sugar in, it's going to dissolve even faster. So this is really important for oral dosing. Here's a bit of data. So the bottom curve here is for micronized orally dosed formulation. So this is plasma concentration over time, and we can see that there's a lower Cmax for the micronized formulation versus the nano size formulation, the nanomedicine, the top curve. So by nanoformulating, we're increasing the uptake from oral dosing. So quickly, let's have a think about how the history of nanomedicine. That was one of the areas I'd cover in my talk. So it's hard to point to a specific start date, but in the 1880s, colloidal silver, essentially silver nanoparticles, as shown on the right, was used as an antimicrobial. Some people have been using it as this man in the middle, as an alternative medicine with some ill effects due to accumulation of the particles in the skin. But actually, we can go back even further than these kind of uses, where Alexander the Great was storing water in silver containers, obviously because it was found to offer some benefit. The talk from Paul Ehrlich around magic bullets was kind of one of the seminal moments around the idea of personalizing and tailoring the way drugs are delivered to target specific cancers in this case. And this really is in line with the very concept of nanomedicine to try and tailor the way the drug behaves within the body through the carrier system. The birth of polymer science after World War I in the 1920s is another key, because it created a toolbox to be able to make polymers with defined structures, with defined polymer chain lengths that allowed the formation and the functionalization of many of the nanomedicines. IV iron that came out in the 30s for the treatment of anemia showed that it was possible to change the route of administration for some drugs by using nanoformulation. So rather than oral, it was possible to switch a formulation that could be administered by IV. Finally, I think another key aspect that happened in the 70s was the wider development and commercialization of nanomaterial characterization tools. So the ability to characterize nanoparticles in their dispersed phase, so in water, for example, and do that on large scale allowed more investigation of the structure and property relationship of these materials. So here's a bit of information on some of the approval dates of modern nanomedicine. So Doxor started in 1995, and my colleagues from the British Society of Nanomedicine will be picking up and looking at more recent examples. But I just want to focus on two of the early ones. So Doxor, the first that came out in 1995, and Rampimune, which was one that came out in 2000, just to showcase some of the ideas. So let's look at Doxor first. Doxor was formulated is a formulation of doxorubicin, the structure you can see in the lower right, which is a drug that was approved in the 1970s. It's a first-line anti-cancer drug, but it's highly toxic. And the ability to use a nanoformulation to improve and change its biodistribution was really an opportunity to change some of the toxicity. One of the big issues is around cardiotoxicity, where there's a dose dependency, and at higher doses that patients have been exposed to, eventually cardiotoxicity is developed. So how can that be avoided? Well, Doxil, which is a liposomal formulation. So liposomes are shown in the bottom left. It's this phospholipid this lipid bilayer structure, and inside it has a crystal of doxorubicin. And you can see on the right, there's a TM image of the crystals inside the liposomes. On the surface of the liposome, it also has a polymer called polyethylene glycol, which helps to alter the way the body recognizes it. So this allowed the drug to be encapsulated and essentially contained within there until it accumulated at the site. In terms of the way it's actually behaved, that ability for it to accumulate in tumors that had leaky vasculature, so the particles would tend to passively accumulate in those sites, led to improved patient outcomes. So here we can see that on the top right, we've got improved survival for patients versus a comparative treatment in topotecan. And a big part of this is the way the liposomes altered the pharmacokinetics. At the bottom, we can see this is IV dosing. We can see the free doxorubicin in the dark color, and we can see that it's rapidly cleared, whereas the Doxil in the liposomes is much more longer lasting in terms of circulatory time. This allowed the drug to accumulate more in the target site. So my final example is about improving the PK. So Rampimune, there's a mistake here, came out in 2000, not 1995. Rampimune is a formulation of rampamycin, an immunosuppressant drug that's used to treat patients with organ transplant. It's practically insoluble in water, so the original formulation had very low bioavailability and used solvents to form an oral solution. What was done in terms of formulating it into a nanomedicine was to mill the particles up down to about 400 nanometers, as shown in the bottom right. And a milling is essentially just grinding them down. And by doing this, the the nanomedicine showed an improvement in bioavailability of 21%. So this allowed it to be orally dosed as tablets, improving the ability to actually have supply chains of the formulation and store it. Okay, so hopefully I know that's been a very whistle-stop tour through the history of nanomedicines. The key message is, just to get started before my colleagues pick up, is that nanomedicines are very different from traditional small molecule medicines. They can alter the PK and the biodistribution of therapies. And I think a key thing to recognize is this area is only growing. We can see in this graph in the bottom right, this is data on the x-axis and number of applications to the FDA. We can see that the number of applications are consistently increasing over time. And there's now more than 50 approved systems on the market and many more in development. So thank you very much for your time, and I'll pick up questions at the end. Thank you.